When we talk about biosimilars, I've always looked at it this way, that generics are for small molecules, biosimilars are for large molecules, and effectively they are both in inverted commas generics, but you don't use the word generic for biosimilar because you can't replicate the product exactly. Whereas a small molecule is small by a factor of I think a few hundred times at least, are smaller. So you actually replicate everything to do with the small molecule, and with the large molecule, obviously it's a lot more complex and you can't guarantee everything is replicated. You just need to make sure that the way that the drug works is in the same way. That's my crude understanding. Is that a fair reflection?
It's not far from from the truth or from the reality. For small molecules, in fact, we're talking about generics because what you have is an exact copy. So you have the same molecule structure and you can really hit it 100% exactly matching. Whereas for biologics, first of all, as you pointed out, they're much larger. So we're talking about 3,000 Dalton, whereas a small molecule can be a few hundreds. So it really can be a factor of 1,000 or 100 of difference in size. But it's not only the size, it's also that there's more variation to the molecule possible. Take aspirin. An aspirin manufactured no matter where in the world, it's always the same chemical formula. When you take biologic, like a monoclonal antibody, your originator product is already a mixture of many, many subspecies because there is so-called sugar side chains that are different. So there's some sugars difference. There is charge heterogeneity. There is some truncated forms. Because the molecules are so big, they're also prone to a step-by-step degradation and modification. So what you actually have is a mix of tens of different sub-forms of your molecule. If you want to make sure that your biosimilar matches the originator in terms of safety comes first, doesn't harm the patient, and efficacy, you have to prove that because you can do so much analytics, but there's still things that you might miss. So you have to repeat clinical studies. You have to really show that your molecule works. Whereas for generics, you have to show it looks the same chemically and similar byproduct pattern, similar formulation, and then just you go without even having to go through clinical trials. That means human tests.
Because a biosimilar doesn't mean it's an inferior product. It could actually even be a better product than the originator because they're never going to be identical, but you could have a better performance with the biologics in for treating that particular indication.
So if I improve, for example, the manufacturing process and I have a higher purity and better cost of goods, it might not be a biosimilar anymore because it might perform a bit different in the preclinical and clinical tests. And there's a concern, and then you have to regard it as a new product. And that's why often we speak about biobetters, which is same mechanism of action, and it learns, if you will, from the originator product, but it's a new product that is built on the knowledge of the initial one. For real biosimilars, it's even frustrating because take Humira. Humira has been developed decades ago, and if I would do a Humira biosimilar today, I would use a totally different manufacturing process, or I would like to, because it's much more performant, I have much lower cost of goods, but I will not match the charge heterogeneity, I will not match maybe the glycosylation, and then it's not a biosimilar anymore. So I have to use a non-performing process or an old-fashioned process eventually in order, suboptimal process, in order to get close to the originator. It's always the gold standard is what has been developed 10, 20 years ago, and you have to match it.